Good morning students, welcome to Leela's tutorial. In last class we have discussed about the physical transfection methods. Today we will discuss about the chemical transfection. So transfection is nothing but transfer of the desired gene into the animal cell. So first we will see what are different types of transfection or methods of gene transfer by using the chemicals. So first one is calcium phosphate precipitation technique. So in this you are going to use calcium chloride solution and the phosphate buffer. So in the first one it is a calcium chloride solution what you are taking, and in the second beaker you are going to take the phosphate buffer in which the DNA, the desired DNA which you have selected is suspended in this phosphate buffer. So now you are going to mix both the solutions and allow the co-precipitation reaction to occur. That means here the DNA which is present inside the phosphate buffer and the calcium chloride will undergo precipitation process to form a precipitate. That is nothing but DNA calcium phosphate precipitate. Right? So now the precipitate consists of the DNA, the desired DNA, and calcium phosphate precipitate. The precipitate will hold the DNA which is suspended in this solution. So now once you get the co-precipitation or the precipitate, now you are going to add this precipitate to the beaker containing animal cells. So now you can see by means of phagocytosis the cell will engulf the precipitate. The complex which is formed, the co-precipitation complex, that will be engulfed by the cell. So as a result you can see there is transfer of the desired gene or the desired DNA into the nucleus of the animal cell. That is called as integration of the gene into the genome of the animal cell. Is that clear? Integration of the gene, that is the desired gene or the DNA, whatever you are taking. Integration of gene into the genome of the animal cell. So from where you have taken this DNA or the gene? From the desired animal, the characteristic, the beneficial characteristic which is present in the animal, you have selected that gene and you have isolated the gene, and then that gene or the DNA fragment must be integrated or introduced into the animal cell. So this is the first method where you are using precipitate, calcium phosphate precipitate, to allow your DNA to get integrated into the genome of the animal cell. Right? Then in second case, it is the cationic polymer. So here for the transfection process, you are making use of cation. Right? So here you are making use of cationic polymers. Here first you are going to take the DNA which is negatively charged, and you are going to add it to the cationic polymer which is positively charged. Right? So here due to the charge, here you can see the DNA and the polymer, the cationic polymer and the negatively charged DNA will form an polyplexus. Polyplexus is nothing but DNA cation polymer complex. Right? Simply we call it as polyplexus. So these polyplexes, due to electrostatic interaction, they go and bind to the surface of the animal cell. Once the polyplexes are formed, right, once the polyplexes are formed, you are going to add these polyplexes in the container having or containing the animal cells. So here the cells which have formed the polyplexes, right, the polyplexes or the complex which is formed is added to the cells. So due to the electrostatic interaction, these polyplexes will go and bind to the surface of the animal cell. Once they bind to the surface, due to endocytosis process, right, due to endocytosis, here you can see the complex is entering into the cell. As it is protruding inside, it forms an vesicle or a membrane which is nothing but your endosome. Right? The particle or the endosome acts as a vehicle and it carries this complex to the nucleus of the animal cell. then it is integration of the desired DNA into the genome of your animal cell. So this is the another mode where you are going to transfer the desired DNA by using your cationic polymer. Coming to the third one, it is liquefaction, or you also call it as lipid mediated transfection or liposome mediated transfection. So here you can see this is the lipid bimembrane, right? So or the lipid bilayer, where you have the head and the tail region, hydrophilic heads and the hydrophobic tails. So that is their property. By using that property, what you are doing, you are taking the lipid bilayer and you are adding DNA to it. So when you add that, they result in formation because of the hydrophobic and the hydrophilic nature. They try to form an vesicle-like structure, that is nothing but your liposome. So during this formation of liposome or the vesicle, the DNA will be trapped inside, and this liposome or the carrier carries that to the cell, animal cell. So now when you add this liposome to the container containing the animal cells, it will go and bind to the membrane, right, and it enters, and this vesicle helps your cell to get inserted with the desired DNA into its genome, right. The vesicle will enter through the membrane, right, through the plasma membrane of the animal cell, the vesicle carrying the DNA. Now you call it as vesicle or the carrier vehicle, right. So the liposome will enter into the cell and it passes through the cytoplasm and it carries the DNA to the nucleus of the animal cell. Then it is the integration of the desired DNA into the nucleus or into the genome of the animal cell, right. This is the third method. Then in the last method, again here also you are making use of lipid molecules only. So a membrane fusion, right. So here the membrane is fusing with the plasma membrane of the animal cell, right. Let us see that. So here also you have lipid bilayer and you have the DNA. So here it is forming an fusogenic liposome, right. The liposome is having ability to fuse with the layer of the outer plasma membrane or the lipid layer of the animal cell, right. So here you can see the fusogenic liposome or the fusogenic vehicle carrying the desired DNA. Now when you add these fusogenic liposomes to the solution containing the animal cells, they go and bind to the animal cell, right, and here they are not forming any vesicle or they are not entering into the membrane. What is happening here? The vesicle, right, the liposome itself is getting merged, is getting fused to the membrane of the animal cell, right. So two membranes. Now you can see here, this is the membrane of the animal cell and this is the membrane or the liposome, right, the lipid membrane. Okay, it is not forming vesicle like this or it is not entering into the cell. But what is happening? The cell or the lipid bilayer itself is forming and continuous, right. The two lipid membranes are merging and forming an continuous layer, thereby throwing all the content present, the DNA present in it, into the cytoplasm of the cell. Then the DNA will go and get integrated into the genome of the animal cell. Is that clear? So here you can see this is the membrane of the animal cell, whereas this is the fusogenic liposome layer. Is that clear? So these are the four types where you can transfer the desired DNA into the animal cell by means of calcium phosphate method, cationic polymers, liquefaction and membrane fusion, right. If you have any doubts, put it in comments. So in next class, we'll discuss the biological or the viral mediated transfection method.